left off, we're on chapter 20. We're talking about recombinant DNA techniques. And I wanted to start with PCR, the polymerase chain reaction. Okay. So, many of you in here have done it at least once. Who has never done a PCR reaction? All right, so, like, many of you, that's just funny. The person who raises his hand does it every week in my other class. I've never done it. Even in general bio, you usually just do it once, if nothing else, right? So what do you guys know about it? Like, why was it so revolutionary to science? What do you know about it? That's so cool and great. Andy, do you want to say something?
Gives you like a higher end.
Yeah, you get such a yield, right? Such an incredible yield in such a short amount of time, a couple hours, 3 to 4 hours, you can get 20 million copies of your DNA in one strand. Freaking amazing. What else?
You only need a very small amount of it.
Correct. You can start with really a small amount and then get huge amounts of DNA at the end. Does anybody know anything else? Brianna, anything? Well, what's a disadvantage? Does anybody know a disadvantage? I mean, we learned about making multiple copies, like through a plasmid or whatever. Rosemary, do you know a disadvantage at all?
It's possibly
Yeah. Yeah, grow up some bacteria is cheap, right? Just give them a little bit of food and put them in an incubator. You don't have to buy all those enzymes and primers and whatever. That's true. It's definitely not as cost effective, though the prices come down incredibly. What about anything else, Andy? Do you think it's a disadvantage? Depends on well you know the technique.
Don't like the ends.
You have to worry about the ends. If you want to, they usually have letters attached in the end, and that's very good. And if you need to, you have to cleave those off. So there's another little cleanup step involved. We had to go in the lab and do that with the endonuclease that we added. Right. So that's like a problem, but one that's been fixed. Like there's a way to fix it. Does anybody know anything else? You can't use it for large pieces of DNA. It's definitely size dependent. So smaller pieces it's good for. And anybody else want to say one more thing before I say the answer?
Develop specific timing sequences.
Oh, yeah. Yeah, that's what I wanted to say. Exactly. You need to know something about the sequence. So if you don't know anything about a piece of DNA and you want to make many copies of it so that you can work on it and research it further, then you can't use PCR. You've got to go back to some of the other methods. But if you know something about it, then you can utilize it. So you do have to make primers. You can either buy them or have them made or make them yourself, and those requires knowledge of the sequence. So that was good that you guys knew quite a bit. So, I remember PCR. Well, I was always a science student, but I might have mentioned it last week, that OJ trial. O.J. Simpson, if you don't even know what that was about, you can read about it. But anyway, that they used this PCR and the DNA as evidence in that trial, and it was a big thing, just like how they utilized it. And now, of course, it's just commonplace. We do it in high schools. All you need is the correct stuff. So it's very fast, right? And you can make many, many, many copies from starting with just a small amount, okay? So some of the things that you need. First off, you're going to need primers, as Rosemary mentioned. That's because DNA polymerase cannot start the strand. It needs a primer there, and then it can elongate the strand. So you need primers. And you need 2 sets of primers. You need one, because remember, polymerase is working from the 5 to 3' end, right, making the new strand. And so you need to have the primers attached here and primers attached here. So those are 2 different sequences. They're not identical sequences, so you've got to have 2 sets of primers. We say a forward and a reverse end. You need DNA, right? So you start off with double-stranded DNA in your tube. You're going to need the enzyme DNA polymerase. So first you need primers. You need the DNA. Sometimes they call it the template DNA. That's the DNA that we're going to utilize and that we're going to make multiple copies of. You're going to need the enzyme polymerase. Often these enzymes need cofactors to help them work, and one of the cofactors that's very common is magnesium. So we need a magnesium present, and we need DNTPs. If I ever told you what DNTPs are, I can't remember. All right, so it's deoxynucleotides, but when you write NTPs, that means it's a combination of all of the bases. So we're going to have in this, you're going to buy it, there'll be ATPs, GTPs, TTPs, and CTPs. And remember, for DNA replication, the energy comes from the phosphate of the nucleotide. It's a trinucleotide, and two of those phosphates come off, and that's the energy. So when you're done, you just have A, MP, G, MP, T, MP, and C, MP. So this is a mixture of all of these three. When I first did PCR way back before you guys were born, you'd have to buy the A's, the T's, the G's, and the C's separately and add them each to your tube. But nowadays you just buy a mix of it. Okay. And so the first thing that's going to happen is that, well, this is actually the first thing that happens right here, is that the DNA becomes single-stranded. It gets denatured. Then the second thing that will happen is that primers will anneal, and then the strand is elongated by the polymerase. So there's three major steps. It's written in the next thing. Denaturing, annealing, and elongation or extension. And there are actually different temperatures that are used for these three steps. And the machine that's used is called a thermocycler. Now in the old days, I had a thermocycler when I started, but before me, when people started, when Carey Mullis came up with this, they used water baths at different temperatures. And they had to move their tubes from one water bath to the next water bath to the next water bath for hours, timing them for like 45 seconds or a minute, and the next water bath, 45 seconds, and the next and the next. So it was laborious. Someone had to be there all the time moving the tubes from one place to another. And of course, each old strand serves as a template. So this is the basic concept. The original DNA is pink. The new DNA is going to be purple-blue, okay, just so you can see that. So here's your DNA to be amplified. And then the first thing that happens, it has to be denatured. And when you denature DNA, you make it single-stranded. And they utilize a high temperature in the 90s. You should realize it's in the 90s. Okay? I want you to know the temperature. That pulls the two pieces of DNA apart. Then the temperature is brought down somewhere in the 50s to 70s. This is putting it kind of low, but all right, let's just say 50. We'll say 50. Okay? For the annealing, for the primer temperature. Now, depending on your primers, you can get different primers that utilize different temperatures, but it's a lower temperature. Okay? And again, you have two sets of primers, one in each direction. And then the extension is back up, let's say, in the 70s. Okay? So 90s to 50s, like basically like the weather's been here lately. Okay, with 20 degrees swings in temperature. Okay? And so what happens then, from that one strand that you had, you now, at the end of after extension, you have two strands, right? One made up from the original template and then the new one. So now those two strands undergo those same steps. First denaturing, then annealing of the primers, and then elongation. And so now when you're done, you're going to have four strands. And it continues on. The number gets doubled and doubled and doubled and doubled each time. Okay? So you should understand this basic process. So the enzyme that's utilized, this isn't even written here at all. Nope. The enzyme that's used is DNA polymerase, but it's not just any DNA polymerase. It's not your DNA polymerase. It's TAC polymerase. So what's so important that we use not your polymerase, not E. coli polymerase, but this Taq polymerase? Why is this so important? Yell it out. What was that? That's right, Brianna. It is thermal stable. This polymerase it comes from bacteria in the hot springs that can withstand temperatures over 100 degrees Celsius. And this is what makes this process, this technique, so easy. Because when they first started doing it, they didn't use Taq polymerase. They just used whatever polymerase that once they hit the higher temperatures, it became denatured and inactive. And so for each cycle, not only do they have to move the tubes from one temperature to the next, they had to add more enzyme every single time because it was completely denatured and useless. So you need the high temperature to denature the DNA, but by doing that, the enzyme was denatured. So once Kary Mullis just was driving along on some stony trip that he was having, because he's a big druggie, going surfing or whatever in California, it hit him that the hot springs bacteria live, and they were able to replicate, so their polymerase must be stable in these different temperatures, and that's how he revolutionized this whole technique. Right? Kind of crazy. When you read about his life, you're like, that's why you just never know who's going to figure something out. You cannot tell at all. So that's the cool thing about it. Okay? Are some limitations, as Rosemary mentioned. You do need to know something about the DNA since you have to make the primers. Another thing, if you, by mistake, got your cells in there, some skin cells sloughed off and got in there, that DNA could be utilized and replicated. And again, long pieces of DNA, this doesn't work well for. But as they say, it is considered one of the most widely used techniques in molecular or recombinant DNA technology. It's used for, it's not just used like in the lab to like make some super cells or something. It's also used for screening of disorders, not just genetic disorders, but medical disorders. I mean, even though medical and genetic are the same, under the same envelope. So that's kind of cool. I really want you to stress this reverse transcription PCR. Okay? And that is, in order to do that, you start off with a strand of messenger RNA. And you utilize that enzyme called reverse transcriptase. We talked about that last time. And then you make this cDNA, and from the cDNA, you use PCR method. Okay? And there's also what's called real-time or quantitative. I don't know if you've heard of this one, PCR. And in real-time or quantitative PCR, you're actually really able to quantify how much product you have in the sample. So this can help you with in medicine and diagnostics, like how much of that bacteria was necessary to cause those symptoms in that person or whatever. So that's the real-time PCR. The quantitative and real-time PCR utilizes, in this case they're talking about TACMan, which is a probe that has, a patent. But basically, there's fluorescence involved, and every time a new strand is made, it gives off a little fluorescence, and the machine reads that. So there's two different strands. This one gave off more fluorescence, so hence there was more DNA and more gene expression, let's say, than this lower one. We actually have an RT PCR machine, and it's not easy to use this measuring fluorescence. I don't know, did we do RT PCR when we were together, Phil? Yeah. Well, for a while, the PCR machine was broken. Wasn't it broken? Yeah, like I went in there and somebody had been cost Dr. Schaefer like $5,000 to fix it or some crazy amount. It was insane. And I'm just afraid to touch it now. All right, but just to go back to this reverse transcription, you isolate messenger RNA. And a lot of times, the way that you would isolate messenger RNA is that you would use a column, and the column would have these little beads, and you'd put the cell lysate through the column, and what you attach to the column would be T's. This is supposed to be the letter T. And then if you take a mishmash of all DNA and RNA and you put it through a column, it has little beads that have T's on it, attached to them, then the poly A tail will bind to the T's. So everything else will come through the column. All of the transfer RNA, the ribosomal RNA, the small nuclear RNA, the DNA, and the messenger RNA that's mature with the poly A tail will stick. And then you would just like maybe change the salt concentration and then elute from here messenger RNA. So does everybody understand how you would use a column first? With beads. I just want to write this down so that you will have it, so that if you had to ever write it for me, this is what I want to see. So the column is beads that have T's attached, and then the messenger RNA will bind via its poly A tail, and then you can isolate the messenger RNA. Does everyone understand it? So now you have a pretty pure amount of messenger RNA. And from there, you can then make the cDNA. And to do that, you utilize the fact that the A's are there. And you make a primer of T's. It binds to the poly A tail. You create that DNA RNA hybrid. They're bound together right here. And then you add RNase, which will break down the RNA, leaving some pieces, though, because you do a partial digest, so it's not completely eliminated. Those are now the primers to make another strand of DNA so that you will have double-stranded complementary DNA. And now you can use this in PCR. Well, I guess you need a ligase. So just let you write that down in case you had to write down the steps of how to start with messenger RNA and then utilize PCR to get the DNA. And remember, what's advantageous of you doing reverse transcription PCR is by isolating the messenger RNA, we know what genes are being transcribed or expressed at that time. So it's a very good indication of what's going on in the cell at that exact time. Any questions? Quantitative, again, is used a lot of times in medicine. All right, so continuing on, let's talk about a restriction map. All right, so, I mean, we talked about libraries, all different kinds of libraries. Restriction map is just not, and it's like mapping genes, but instead, on a piece of DNA, you're mapping restriction sites. Okay, and you know that the restriction site A, T, U, oh, no, U, sorry, C, G. Oh, boy, A, T, C, G is right here, and so forth. So that's what's called a restriction map. And it's important if you're going to utilize your DNA for other studies to know where these maps are, or to know where these sites are. With a plasmid that's been genetically engineered, you know where the restriction sites are because you ordered it with given restriction sites. Okay? We utilize the fact that DNA, or our DNA, has all these different restriction sites in all these different places to help us tell my DNA from your DNA, from her DNA, from his DNA. So restriction mapping is very, very common. And then you run your fragments after you cut them with restriction enzymes. You run them on an agarose gel via electrophoresis. The smaller pieces move faster and go towards the bottom of the gel. The larger fragments go slower. They stay towards the top. And then you need some way to visualize these fragments so you can see. So this picture coming up is utilizing ethidium bromide, which maybe a lot of you have used ethidium bromide to look at gels, because this looks familiar. Didn't I show you a hard copy of a gel in here? Was it this class? The other one? So it was just a hard copy. I've been carrying it around, but I still have it. Yeah, of some gels. So you guys in the front saw this already, right? All right, give it to Tim when you're done. It's just a hard copy. So each one of these little lines or little bands here represents a piece of DNA that has been cleaved or cut with restriction enzymes. So if this was your big piece of DNA, the enzyme cut here, cut here, cut here, cut here, here, wherever the restriction sites are, and gave you all these different fragments, right? And then in the top of the gel are the wells, and then you would take all the fragments and add them there, and then you have electrical current that runs through it, and the fragments run. The smaller ones go towards the bottom, and the bigger ones stay up on the top. So with this kind of picture, it's like the picture I'm showing you. That's an X-ray of this type of picture. They utilize lithium bromide. We don't use it as much anymore. It is much more sensitive than any of the other methods, other dye methods that we use, but it is like a neurotoxin, so they try not to utilize it as much. And one thing that's blatantly wrong with this picture, guys, look how the ruler is. Like, nobody measures DNA this way, right, on a diagonal basis. It's always how far does it go from the well and how far did it travel, right? And that's how we tell the difference in the base pairs. So maybe it looks good in the picture to have them, but I want to say that's so wrong. I just have no credit for your textbook for putting in the picture with a ruler that way. Okay, so these are smaller pieces and these are bigger pieces. And when you look at this, you almost get this quantitative feel, right? Like there's way more color here than there is here. So there must be more DNA here. Probably true. Probably true. But this is not a quantitative method in any way. It's just to show you the base pairs. Okay? And this is how we can tell you from me, is by taking my DNA and your DNA, same pieces, using the same enzyme. And so this is you, and you're going to cut, and you're going to get 1, 2, 3, 4, 5, 6, 7, 8 fragments of these various sizes. When the enzyme is used to cut my DNA, it cuts here, it cuts here, it cuts here, it cuts here, and it cuts here. And I get 1, 2, 3, 4, 5, 6 fragments, and they're of different lengths. So the banding patterns will look different. Okay, and that's one way we can tell one person from another. I'm putting it very simplistic, in a very simplistic manner. When you see something like this, what does this kind of a band tell you guys? You know the answer. When you get it like that, it's so much. What does it tell you? There's so much DNA there, right? There's so much, it's like smearing and so much. No? Whatever. Okay. So after you get your run your gel, right? You can then, with DNA, you can just take a picture like this. You can take an X-ray like the picture I'm showing you, like that's being sent around, and you can have that X-ray film gives you a hard copy. There are other ways to make a hard copy. So that's where the southern blot, the northern blot, and the western blot come in. In a southern blot, you have run the gel, you've run your DNA fragments on the agarose gel, and then you transfer those fragments to a piece of nitrocellulose. So southern is for DNA, and that gives you an actual hard copy on that nitrocellulose. And then you need to probe or hybridize. Remember we talked about this word before? A probe, which would be complementary to the DNA that's on that nitrocellulose. The probe could be DNA or RNA, and it usually has a color or radioactivity associated with it. Okay, so that's a southern. Here's a picture of that concept. Here you put your DNA up in the wells, different people, let's say, or whatever, and now you run your gel. This is what it looks like with all the little fragments. Like this first lane is considered the molecular weight markers, and then here are the two different people or the same person, whatever. Then you do the southern blot, where Down here is the gel, and on top of the gel, oh, no, it's right here. Down here's the gel, and on top of the gel is that piece of nitrocellulose. And through capillary action, the DNA migrates from the agarose gel and gets trapped on the nitrocellulose. And then you put the nitrocellulose truly in a Ziploc bag, you add your radioactive probe, let them bind, and the probe will bind where it should be, and then now we can read that gel. It's a hard copy. What's really great about this is that now you have this hard copy that has all this DNA on there, but your probe was just to a specific sequence. So the probe bound over here, and over here, and here, and here, and here, right? But all that other DNA is still there. So later on, next week, next year, 10 years later, you can remove the probe and reprobe it with something else because you have a hard copy with your DNA right on that nitrocellulose. The agarose gel can't withstand the test of time, but this nitrocellulose paper can. It's not uncommon for somebody to go in someone's notebook from years ago, pull out a southern blot, northern blot, or western blot, and reprobe it for something else. So it's kind of cool. And this creates a hard copy, a truly, truly hard copy. Not just a picture hard copy, but the actual hard copy of the gel. It's great. So if you do it with DNA, it's called a southern blot. So this is the concept. This is all the DNA that's on that gel. But after you probe it with a specific probe, these are the bands that light up that your probe bound to. Next week, you could do a different set of probes, and you'd get different lines, let's say. So it's really neat. A northern blot is the same concept, except you're using RNA. So what's on your gel first is RNA, then that's transferred to nitrocellulose. And a western blot, you have proteins. You run a gel with proteins and then transfer to nitrocellulose. Dr. Southern was the one that figured this out. He started with DNA, and then they thought, funny, haha, let's make a northern. It's exactly opposite of DNA, and that's RNA is opposite of DNA, and then they're like, oh, funny, ha, 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 let's make a western. It's not really related, but it kind of, and blah, blah, blah. So scientists are just so funny. You should know the difference between a northern, southern, and western, what they're looking at. There are other ways that we can see our sequence that we're interested in of DNA, and one of those ways is the fish method. Has anybody done this? Did you do this in cytological techniques? Fish? Okay, so I didn't know. So this means that you're actually going to use a probe. It's usually a colorimetric probe in situ. That means on a living organism, on the cells. Okay? What was that? On people, yeah. Oh, in the paper they utilized it. Yeah, okay, I didn't know that. So, yeah. So like in embryos or something like that. So this is just a picture in your book. Colors, like the purple color is where the probe bound. So whatever you're looking for was right at that area. So if you have a probe to a specific gene sequence or something like that, you can see where it may be in the embryo. It's not, I'm not telling you much about this picture, but it's pretty. That makes the book. Okay. The very last thing I want to go over is DNA sequencing, how to find out the actual letters of that code. And remember, DNA Day was just April 25th, where it was 15 years, right, since the human genome was sequenced, and it took 15 years, but remember we learned that if they did it the old-fashioned way, it would have taken 8,000, right? Did we talk about DNA sequencing already in this class? Or was it the other class? The molecular, sorry about that. We'll get those two classes mixed up when I get to this part. All right, so the traditional method is the Sanger method for DNA sequencing, okay? And then they took the Sanger method and they added a contemporary twist to it, which automated DNA sequencing and made it happen faster, okay? So the difference here is that they're using a dideoxynucleotide instead of a deoxynucleotide. Normally, we have deoxyribonucleic acid, right? And it has a hydroxyl group at the end of one of the sugars, at the end of the sugar. But a dideoxy, the oxygen has been removed, and it's just a hydrogen. Let me show you. And when you have a dideoxy, you can't extend the DNA strand any further. So this is what it typically looks like. Deoxy with and carbon number 3 has a hydroxyl group, but the dideoxy, it lost the oxygen, it's just a hydrogen. So there's no binding here. You can't make that phosphodiester linkage. Okay? So utilizing this idea, this is what happens. So here's your strand that you're trying to figure out the sequence of. You have to have a primer, because of course you need a primer for DNA replication. And then you have, in this case, colored or fluorescent, and this is colored, dideoxy. You have like A's are red, C's are blue, whatever. G's are green, and T's are yellow. And you put a small amount of deoxy, and an even smaller amount of dideoxy. In other words, your normal amount of deoxy, but then a smaller amount of these specialized dideoxy. So wherever a dideoxy A binds, the strand will not be able to be elongated any further. So you can see here with this strand, you had DNA replication continuing, right? And then all of a sudden, a dideoxy C was added. So that's the termination, that's the end of the elongation of that strand. See, before, these two C's were these three C's were the regular deoxy C's, and the strand could be elongated. But once a small percentage of the dideoxy binds, you cannot elongate. So by having a lot of template DNA in here and just keep having many, many cycles, you're eventually going to get strands where every single A will, not one strand, but in all the strands, every single A had a dideoxy. Every single T, every single G, every single C. And so then that's red, it's run on a kind of a gel and utilizing capillary gel electrophoresis, and then there's a little laser in the detector, and it runs through, and the colors are read off. And you get this banding pattern that's like blue, yellow, red, green, red, blue, red, da. They each represents the different letters. And that's how you can figure out the sequence. It's a little bit more complicated, so I wouldn't ask you to write an essay on it, I don't think. That's it's so cool. So in the old days, I should have brought in I have some DNA sequencing images, just like I'm passing around these. I have some from the old DNA. So when they would run these gels in the old days, they didn't use capillary gel electrophoresis. Before they came up with this method of utilizing this fluorescence in a computer, they would use radioactivity. And so you'd have to go back and read the gel like a gel like this. It's like this one. This one here. And you'd read it from the bottom up. So it would be like AT, AAT. So that's how they would do it. So I was in a lab in the 90s where they were doing some DNA sequencing. And so when they would do this, the plates that they would use to run the gel would be like maybe 3 feet by 4 feet. Big, gigantic glass plates. And then they would have 2 pieces of glass with like little spacers, and then a comb goes on the top, like a Western. It's a polyacrylamide gel, but very big pieces of glass. And then they would pour it, and then they would run it, hook it up to electrophoresis. Then they take this gel, they would do Southern, transfer it to nitrocellulose. It was radioactive. And then they would put it against X-ray film, and then they would read it that way. It took them forever, and they had these big-ass pieces of glass that they had to work with. They had to clean them meticulously. It was just a lot of work. And every time I looked at somebody doing it, I was like so happy I wasn't doing it. But then, some people all over the country and the world were involved in the Human Genome Project. So all these little labs, people were just trying to sequence, and they all came together to make the genome. Anyway, I was not involved in that. I was thankful all the time. So this then became computerized and gave us large amounts of sequence information really quickly. So in this case, they use this little molecule here that's like anchored onto a substrate, and when the strand goes through, it gives off fluorescence, and that's read by the computer, and you get a strip that tells you the sequence. So it's, again, the same idea of a fluorescent tag, but it's all automated. No gels, nothing like that. Cool. This is just interesting on how much money they spent. This is dollars per, what's the M stand for? Kilobase, not kilobase. Is it mega? Right? Mega base. So how much they spent, and you can see that's in green. They started to spend so much money. And then as the automation got better, right, they spent less and less and less. Right? And, you know, of course, they progressed very quickly with E. coli was the first organism to have its genome completely sequenced. And then slowly came the yeast and the humans and the mice and so forth. All right. I said that was going to be my last thing, but just, I don't know how much you guys know about knockouts and knock-ins. Does anybody know anything about this? At all? Knockouts?
Just knock out a gene.
Yeah, to knock out a gene, like knock you out, KO, hit you down. So through a double crossover, you can basically take a non-functional sequence and put it in where a sequence was, and now the gene is non-functional. It's not even there anymore. You've knocked it out. And then knock in is the exact opposite. Put your sequence in, make it functional. So that's really, we're working on, you're not even working on the cellular level anymore. You're actually working at the level of the gene locus because you're having that double crossover. So you want to say, hey, let's say I get rid of this gene, what's going to happen to the organism? That's really how it all started. Seriously, like, this whole knockout started in the 90s. And they were like, let's just get rid of this gene and see what happens. Let's see if we get rid of the gene for actin. What would happen? And that's what they just did. They just started knocking shit out of all these cells just to see what would happen. So that you can see here, that's you had this double crossover, and of this target gene, you've knocked it out and changed everything up. And they're just giving you this example. And then what they would do is they would find the cells that had the knockout. They would implant those cells into, in this case, a blastocyte, put it into a pregnant female, take her out by giving her another embryo, basically. You know, and so you get a chimera, which is really basically heterozygous, and you breed them selectively until you get pure homozygous for your knockout. So this is how they kind of did it. So this is a knockout, and you can appreciate that a knock-in would be adding something. You can use these in stem cells. You can make fat mice, whatever. I know. They didn't feed this a lot. They just knocked out something. And he just changed his whole metabolism. But you can see more about it. You can even put knockouts and knock-ins to turn on at different times. Because remember that at any given time, 10% of your DNA is being expressed. So if a gene is being expressed later in the life cycle of the organism, you can have it set up next to a gene that's going to be turned on at a different time. And so that's this whole transgenic animals, knock-ins and knockouts. And I already showed you this picture where they made the mice fluorescent by knocking in a gene for green fluorescent protein. Okay, so this is truly all she wrote. Okay, so next time I see you, final exam. No, test number 4, but then final exam. I just sent myself a note for that.